Hi, my name is Samantha Foti, and I'm a quality control scientist at Aldevron's Madison site. Aldevron is a contract development and manufacturing organization. We supply research to GMP-grade Cas9 proteins for therapeutic applications. And in recent years, our clinical partners have also expressed a need for similar research grade to GMP track for CRISPR RNPs. And so today I will be presenting on the work that Aldevron has done to distinguish the conditions around complexing CRISPR RNPs and the development and validation of methods used to test them. RNP stands for ribonucleoprotein, which is a complex made of the guide RNA and the Cas9 protein. The graphic here shows the Cas9 protein with its catalytic nuclease domains, cleaving a piece of genomic DNA after alignment to the guide RNA. The DNA is cleaved on both strands, resulting in a double-stranded break, which allows for researchers to knock out or inactivate a gene inside of a cell. Delivering RNPs inside of a cell is just one way to perform CRISPR gene editing. CRISPR nucleases and guide RNA can also be delivered as plasmid DNA or as RNA. However, the use of RNPs is advantageous for a few reasons. So first off, the RNP bypasses transcription and translation, so it's immediately active. And then second, due to the fact it is a RNA protein complex, the RNP is quickly degraded, which reduces potential off-target effects. And then a third benefit is using an RNP is that they're able to be titrated. So that pretty much means that RNPs can be delivered in a controlled fashion. So there are a few benefits to using RNPs compared to other delivery methods. And now I'll discuss some of the examples of CRISPR RNPs used for therapeutics. One of the leading therapeutic examples is the use of CRISPR RNPs in ex vivo treatments for immuno-oncology. And in recent years, there's been a rise in the development and use of CAR T therapies against certain types of cancer. CAR T is short for chimeric antigen receptor T cell. The first two CAR T therapies approved in the US, Kymriah and Yescarta, take a patient's own T cells, modify them by delivering a lenti or retroviral cassette expressing the chimeric antigen receptor, which then randomly integrates into its genome. And then this receptor targets CD19 on the cancer cell surface. Since the approval of such therapies, there have been a number of studies performed that show when endogenous T cell genes are knocked out, it can greatly enhance the therapeutic potential of CAR and TCR-based therapeutic approaches. So CRISPR RNPs are the safest and most effective way to inactivate these primary T cells. Some examples of endogenous T cells that can be targeted for knockout to create a therapeutic benefit can be seen in the table below. So we have listed first are examples of native T cell receptor genes. When these genes are knocked out, it prevents cross-reactivity with the exogenously delivered T cell receptor. And then next, the PD-1 gene, which is involved in T cell exhaustion and suppression. And lastly, when thinking about universal off-the-shelf T cell therapies, we move to HLA or human leukocyte antigen genes. And these are involved in graft versus host response. Knocking out these genes in different combinations to figure out which ones to remove puts us closer towards potential off-the-shelf T cell therapies. And these are just a few of the more recent T cells that have been identified as having beneficial therapeutic impacts, but there are more coming out in the literature every day. Aldevron recognizes that there's a need in the field for therapeutic RNPs to complement our clinical GMP-Cas9 offering. So in collaboration with our therapeutic partners, we first set out to determine the ideal complexing conditions for guide RNA and Cas9 protein. Next, we wanted to optimize a platform in vitro cutting assay to assess the relative RNP activity. And then another aim was to develop and optimize assays for quantifying free or uncomplex Cas9 in guide RNA. And next, we created a standard QC panel for RNPs, as well as conducted long-term stability studies to determine shelf life and optimal storage conditions for RNPs once they've been complexed. The poster on the left is a culmination of the RNP work done at Aldevron. So our first task was to optimize an in vitro cutting assay to assess the relative cutting activity of each RNP. We used a linearized 5 kb plasmid, which contained the guide RNA target. And then if you look at the gel on the top left, the first lane next to the target contains 3 bands. The top band represents the 5 kb DNA target. And then once the DNA target is cleaved, it creates asymmetric 2 kb and 3 kb bands. If cutting takes place, you expect to see a diminishment of the top band and an increase in intensity of the bottom 2 bands. And as we move from the lanes left to right, there is a decrease in the amount of RNP tested, and with fewer RNPs, there's less cleavage, hence the increased intensity of the top band of DNA. The dilution scheme was a critical assay to set up because we wanted to figure out the linear range for cutting for a particular RNP, as well as the lower limit of detection. As you can see here, the limit of detection based on the gel's densitometry is around 0.01 picomoles of RNP. And we were also able to find a sufficient linear range of cutting as represented on the graph on the top left. Also, it's important to note that these gel images are representative of the assay for one RNP. So there have been similar additional studies done in which the data is not depicted here. Once we developed a satisfactory in vitro assay, we worked on discovering the optimal amount of guide RNA to protein ratios for the RNP complex. Using the gel cleavage assay, you can see in the far left lane of the bottom gel, a non-template plasmid control. And then in the second lane, there's a linearized DNA target plasmid without any RNPs. These samples don't contain a target site, so we wouldn't expect to see cleavage. They just have that single band with no bands below. And then we looked at ratios of 0.5, 1, 1.2, and 1.5 times guide RNA to Cas9 protein, and looking at a titration of the RNPs for each of these ratios. Based off this data, we concluded that the 1.5 times ratio was able to achieve the efficient level of cutting in vitro. And these results were also substantiated in cellular models whose data will not be presented today. But in conclusion, we were able to develop a nice in vitro cutting assay for RNPs, as well as identify the ideal complexing ratio of guide RNA to Cas9 protein using this assay as a guide. And next, we focused on quantitatively measuring free Cas9 in the RNP preparations. So we tried a couple different methods before finding the one that worked best. Initially, we tried to use differential scanning fluorimetry as a way to measure free or apo-Cas9, and this is represented by the graph on the left. However, we weren't able to find a linear range, so we then turned our attention to size exclusion chromatography, which is represented by the graph on the right. And the gray line shown here represents the RNP, and it elutes around 6 minutes. But unfortunately, so does the free Cas9, making them indistinguishable and the assay unfit for purpose. And then lastly, we looked at using analytical cation exchange chromatography in order to differentiate between the bound and unbound Cas9 in the RNP composition. On the far left is a graph representing the free Cas9 by itself with no guide RNA. And on an analytical cation exchange column, the Cas9 elutes between 15 to 17 minutes. And when we compare the RNP on the same cation exchange column, it's shown eluting between 9 to 12 minutes. And also note that the guide RNA elutes in the flow through, so it's not bound to the column. Based off the graph on the far right, it's evident that the free Cas9 in the composition, or the blue line, and the RNP complex, or the green line, are sufficiently distinguishable. And after significant development, it was determined that analytical cation exchange was the optimal assay for determining free Cas9 in the RNP preparations. In the start of our work with RNPs, we noticed that the composition and quality of the guide RNA can greatly affect the resolution of these assays. You can see that on the gel on the left, there are 6 separate guide RNAs, and the first lane next to the latter has a single distinct band representing the high quality guide RNA. There's little to no peripheral products to the band of this RNA. However, in the remaining 5 lanes, we see that there are a number of products in addition to the full-length guide, which represent lower purity guide RNA. And when we tested the RNP samples on the analytical cation exchange column, You can see an RNP ran using the high purity guide RNA results on the graph on the top right. And there's a nice separation between the RNP eluting around 10 minutes from the free Cas9, which elutes just after 15 minutes. And this is based off the properties of the negatively charged guide RNA complexed with the positively charged protein. And then the RNP is overall less positively charged than the unbound Cas9, so it'll elute sooner. And when you have extraneous guide RNA products, which is what we suggest to be present in the lower purity guide RNAs, this confounds the resolution of the cation exchange assay. And we can see the results of the RNP containing the lower purity guide RNA in the bottom right graph, in which the RNP has become indistinguishable from the free Cas9. We determined that these findings regarding the purity of the guide RNA was valuable information to share with our partners who had clinical aspirations for their genome editing therapies when considering the reagents they would need to do this work. Once we established an assay for free Cas9, we directed our efforts towards quantifying free guide RNA, and through experimentation again with a variety of methods, we discovered that native page cell electrophoresis was a useful way to separate the free guide RNA from the complete RNP complex. The gel on the left represents increasing ratios of guide RNA from 1 to 1.5 times RNA to protein. And then this gel was stained with cyber gold to detect the RNA. And then to the right is a Western blot of the same native page gel in which we used an antibody against the Cas9 protein, which is represented by the red staining. And there you can see the staining overlaps with the entirety of the top bands, indicating that the guide RNA is complex with the protein in that region. And we could conclude that the bands on the bottom of the gel represented free guide RNA running further than the RNP complex. And also note, as we increased concentration of guide RNA to Cas9 protein, we see an increase in density of the free guide RNA bands, and this just indicates more free guide RNA is present. So one of the greatest unanswered questions around RNPs was related to the conditions around their long-term storage. Looking through the literature and commercial protocols, there's really little to no data surrounding the longevity of RNPs, and in fact, many RNP complexing protocols actually called for immediate delivery to cells. So if we're thinking about the potential of RNPs as an off-the-shelf drug, it was critical to understand the best storage conditions and the stability over time. In order to find answers to these important questions, we set up a long-term stability study with storage conditions at -80, -24, and 30 degrees Celsius. We placed RNP samples at all 4 temperatures for up to 6 months, and then samples at -80 for up to 2 years. Here are the results from the cleavage assay using samples stored for 6 months. Shown here are gels for all 4 temperatures. This is the same gel-based cutting assay as previously described. And when you look at the -80, you can see there are 2 ladders that flank a negative control where no cutting has taken place. And then in the first lane to the right of the second ladder, we see a low intensity of the top band, indicating a high level of cutting is occurring. And similar results can be seen on the -20 gel. And note that both of these gels are almost identical to the results we saw for time point 0. And as the temperature conditions increase, we see a similar cutting profile with the 4-degree RNP sample. However, we see a decline in cutting efficiency with the 30-degree sample. As we take a look at our 6-month stability study results for analytical cation exchange assay, we see a nice tight peak around 10 minutes for samples stored at -80, -20, and 4 degrees, which indicates an intact RNP with few degradation products. Once again, these results are very comparable to the results of time point 0. But then as we observe the results from the 30-degree sample, we can see there's a complete diminishment of the RNP peak, and this is likely due to the degradation of the guide RNA and Cas9 protein at this warmer condition. This data supports the idea that the -80 up to 4 degrees seems to be a suitable storage condition for the RNPs for up to 6 months. And in contrast, the 30-degree temperature was not a stable condition. The study continued to extend for 12 months for our -80 storage condition. And on the top right, you can see the gel images compared from 6 months to 12 months. Once again, looking at the 4th lane, or the first lane to the right of the second ladder, we see a high cutting efficiency for the 6-month sample. And then as you look at the cutting activity of the RNP after 12 months at -80, you can see there is no apparent decrease in cutting efficiency after the additional 6 months spent in storage. Once again, these results are nearly identical to time point 0 data collected. The analytical cation exchange chromatography graphs also support the activity assay findings, and that they show very comparable results for both the 6 and 12-month time points, with the tight peak eluding around 10 minutes. And it's very informative to know that RNPs can be stored at -80 for up to a year with relatively little degradation or inhibition in cutting activity. And also note that these studies are going on for 2 years, so there will be more data available in the future. At each time point, we also ran our assay for the free guide RNA. Here you can see gel images at 3 months and 12 months. The last 4 lanes on the 3-month gel show RNPs stored at -80 through 30 degrees. These RNPs were formed at a 1.5 times ratio of guide to Cas9, so we expect to see free RNA. We do see a single band of free guide RNA in the -80 and -20 samples, but we see degradation in the 4-degree sample, and then the 30-degree sample shows nearly complete degradation of free RNA. In the bottom gel, we can see the -80 sample ran in triplicate after being stored for 12 months at this temperature, and all 3 lanes are nearly identical to the results presented at time point 0 and 3 months. The tight band of free guide RNA with no apparent aborted products below confirm that the RNP storage at -80 for up to 1 year is suitable. That concludes my presentation. Aldevron's goal is to be the leading supplier of therapeutic material to all of our clients doing genome editing work for clinical application. Feel free to contact Aldevron or me directly with any questions or inquiries on how we can help you move from the bench to the clinic with your genome editing therapies. Thanks so much for your time.